A very good morning to everyone. I, Meenu Agarwal, Assistant Professor in Department of Biotechnology, welcome you all for joining us on the second day of International Conference on Biotrends, Advancement and Challenges in Science and Technology conducted by Department of Biotechnology, NIET. Now, I'd like to introduce our first speaker for the day, Professor Pawan Dhar. Professor Pavandhar is currently the Dean, School of Biotechnology, Jawaharlal Nehru University, New Delhi. Synthetic Biology Group, School of Biotechnology, JNU, New Delhi. Professor Dhar received his PhD degree in 1993 from BHU in the area of human genetics. Professor Dhar has launched and incubated the first biotechnology company in JNU to deliver therapeutic molecules from a novel drug discovery platform. Prior to this, he held senior scientific positions at Shiv Nada University, Riken Genomics Science Center, Japan, Bioinformatics Institute, Singapore, Kew University and Kyoto University in Japan, and Manipal University. In the past, Professor Dhar has headed Riken Japan and TCS Hyderabad program on metagenomics, established research lab in India, Singapore and Japan, and has a total of more than 80 national and international publications in genetics, biotechnology, bioinformatics, system and synthetic biology to his credit. He has industry collaborations with Intel, Bangalore, and Tata Consultancy Services, Hyderabad. Professor Dhar serves on the board of referees for European Science Foundation, Executive Council member, Chair Intellectual Property Management, member Biosafety Committee, Academic Committees of CCMB, NII and Indian Naval Academy, DBT, Task Forces of Marine Biotechnology and Genomic Engineering. He is founding editor-in-chief, Systems and Synthetic Biology Journal, Springer. We are pleased to have you here, sir. Please enlighten us with your thoughts.
Thank you very much, ma'am, for your kind introduction, and a very good morning to all the participants who have joined in this international conference. Before getting started, I'd like to know, this is 10:28, so my time duration would be up to 11 o'clock, right?
Okay, sir.
Is that correct?
Yes, sir.
All right. So thank you very much. I'll be now sharing the screen, so please do guide me. This is the first time I'm using Teams, so can you see the screen? Is it?
Yes, sir.
Yeah, all right, then I get started. So today I'll be talking on systems and synthetic biology, and this is very recently come up in the community. Hardly, the last 1.5 decades, we have been seeing some advancement in these areas. So my intention is to walk through the fundamental concepts, and we'll try to see what was the reason for systems and synthetic biology fields to come into existence, and what kind of advancements have been there for the past few years, and also touch upon our work. So reductionism has been the key scientific approach in doing science. Reduction means that you have an organism and you want to know how it works, how it functions, and then you want to search for an answer within the organism. So you have to go deeper into the organism in order to understand how it functions. So this approach of going from the large scale to a smaller scale is called reductionism. And when you look for an answer, then you come across these organs, these systems that are stitched together to create the whole organism. And now, when you have a reasonably good view at this level, obviously you want to know what's there deeper, at the deeper level, because these do not give you enough answers. So you're looking for answers. So you go to one level down, and then you come across the cell, And the cell, if it is stained in a normal way, then it looks an empty structure with something in the middle. And then you investigate further based on your technology development, and then you realize, and the picture on the right corner, you realize that the cell that seemed to be very innocent is actually full of details. And now your technological aid and staining techniques, they both help you to investigate further. So when you go deeper, people came across this structure called chromosome, and they found that DNA needs to wind itself 6,000 or 7,000 to 10,000 times to make a chromosome, and unwind itself to make chromatin. So this was very is a statistically precise activity, a mega activity at a micro level. So earlier, when DNA was identified as a big molecule and kind of a superstar in the community back in the 50s, people thought of DNA as more of a director of nuclear affairs, that it is the molecule, it controls everything. But over time, RNA community and the protein community caught up the attention. And so over time, what has happened is that DNA has become more of a database of information rather than the regulatory molecules. So now when you go a little deeper from DNA, you come across chemical reactions. Now these are chemical compounds talking to each other. So now you leave typically the biology and you enter chemistry. And if you go deeper, you come across atom. And when you go a little more deeper, then you see that an atom is 99.99% space. And you cannot go deeper because how do you view the space with a microscope or with something else? Therefore, when you're trying to understand biology and you're going to that level, you understand that as you go deeper and deeper, you're losing resolution or the control over information. And so when these atoms talk to each other, when the atoms talk to each other, chemistry is born. And when chemistry comes into biology, and then you see biochemistry and other forms of biology, fields of biology. And so overall, what we have just seen is this process called reductionism. And this reductionism is giving rise to components, and we call them as genes and proteins and RNA, carbohydrates, lipids, and so on. And this particular method of generating data from the cell worked pretty well, and as a result of which we have a large body of data. Now, in the mid-90s, there was a big problem, what to do with this data, because it's a lot of data. So then the era of capturing the data in the form of databases started, and the practice of modeling the data. Modeling means that you're creating a representation of the data in the computer. And so even a database is a model in that respect. So after the modeling, then there was another thought. Early 2000, 2001, this thought came up because people said that now we know how to reduce the information, how to split open the system and get the components. That is one way of doing things. The other way is to capture these components in the computer and do experiments through computers. So that's another way. Could there be another way, the last one, of designing the systems that did not exist in nature, or they exist in nature? You're constructing the systems. So the first approach is commonly called as molecular biology or biochemistry. And the second approach is more often recognized as systems biology, or at component level, you can say bioinformatics. And the third approach is what we call synthetic biology. So just want to get the definition right before we delve deeper into these fields. One is about reducing the information. The second is about capturing the information. And third is about creating a new information, right? So you have three different fields. So the integration of information has led to creating virtual red blood cells. This was the project that I joined in Japan back in early 2000. They published, in fact, late 90s, they published a paper in bioinformatics where they, the group led by Masaru Tomita, for the first time established that one can transfer biological information in the form of pathways to computer, make predictions, and they can be experimentally validated. So likewise, people are now looking at the possibility of making virtual skin that can model the growth of the hair, the color of the skin, and the aging process together can be captured in the same model. And this is a big project in the US and Europe, the Virtual Brain Project, which has taken off very well, and a lot of information has been derived from here. New technologies have come from this project. I'm not trying to oversell the modeling because it can lead to incomplete understanding, which means that the real object is the real gene, protein, system, cell, tissue, organism, whatever you call it. But when you capture it in the form of a computational equivalence, in the form of a database or in the form of a pathway model, then it becomes a representation of the system. It's not the system itself, but it's a representation of the system. And as you can clearly see, the representation of the system can be measured nicely. Say the shadow can be measured quite well by length and breadth. But what has happened is you lose a dimension, right? So information loss is essentially an activity that you come across while modeling the system. So you don't model each and everything in the system. You model only the key components and hope that these key components will represent most of the system behavior. Now that's the art of modeling. Sometimes it can also lead to incorrect understanding. So what you model, what you got in the computer may not match what is there in the real system. So you got to revise the model, you got to revise the values and parameters and so on. So this is the thought I proposed, quite some time back. Is it time to construct a periodic table in biology? And if yes, what would be the components of this periodic table? Because if you have a periodic table representation, accurate representation, engineering of biology gets easy because you start at the molecular level and you can assemble the components and predict what is going to be constructed. So it has a payoff, but then there's a huge information gap above and below the fold level. So this will remain for quite some time. And so the question is, can biologists think like engineers? Why are we thinking like that? Because there are similarities between biology and engineering, as you can see on the screen. However, there are also dissimilarities, especially the fact that if you design engineering systems, you're talking of components that stay there. They don't move all the time. Even if they move, they're glued to something. They're latched to something. So there is a certain firmness to their address or location. While as in biology, you have RNA, protein components that are moving all the time. They don't stay at one place. So how do you hardwire these molecules that are moving all the time from one compartment of the cell to another compartment? They are moving. So you've got to have a different engineering approach of stitching their properties together. Engineering systems are predominantly digital, like the ones that we are using today, the computers. But biological systems are predominantly analog, and we'll see a graph on that. And there is a unidirectional flow of information in digital systems, while as in biology there is a reversible flow. So, for example, as you can see, the enzyme substrate complex. The enzyme substrate complex itself is an indication that some of the information from the enzyme substrate complex goes back to the substrate. So there's a reversibility of the information, which is not found in the engineering system. And Mendel's laws are the only ones that we know. If you call them laws, some people like to call them as ratios, then laws. But at the end of the day, they don't help because they are zoom in, zoom out types. So they zoom in from phenotype to genotype and genotype to phenotype. So they don't help you in designing molecular systems. All right. So in this background, when you got to make something, then you have to consider that your systems are noisy and unpredictable. So noise means there's a fluctuation of the gene expressions at every time point, and unpredictability because we do not know what's going to happen if you remove or add a gene. And if you have to make a biomolecular circuit, the information is lost at many steps, at 2 steps at least. Before the information reaches the next point, it has to go vertically downwards, survive certain filters, and if the information survives, then it goes to the next step. All right, so it's not very, very easy to construct a biomolecular circuit as against the engineering circuit. So To design well-behaved system, one needs standards and rules of composition. Now, the standards are the ones that remain fixed. It does not mean that standards are some God-given features. No, standards are the ones that are made by humans. So, for example, in this case, the standard that was made in the broad gauge, in this particular example, is 5 feet 6 inch. It could have been 6 feet. It could have been 7 feet. Well, somebody fixed it because of some reasons at that time. And then after this, you have a meter gauge, you have a narrow gauge. That also is possible. So standards are something that you fix them once for all. We do not have these fixed quantities in biology most of the times. And then we need to put the molecules together to construct higher order system. Then we have rules of composition. So we compose the system in a certain format. You cannot put the parts together randomly and hope that a system comes up. You have to be very precise in what order do you stitch these parts together. So you got to know the standards and the rules. If you know both, then you can construct biological systems. And the grand challenge is build the chemical synthesis of cells using rational design approach. Now, IEEE standards that are very common in engineering do not exist in biology. Therefore, overall, it is good to use engineering as a philosophy. Then we don't have any tools. So you got to make those tools. The reason they don't exist is because probably engineers never faced a problem. Now, this is not to say that what they are doing is less complex to biology. It is just to highlight that there's an uncertainty, emergent phenomena, probability, gradients, and the amount of new information that comes up. Emergence means that something happened with the same group of molecules that was not predicted before by any computer. It was just not possible to predict it, but something new happened. This happens in biology all the time. Likewise, when the transcription factor talks to DNA, then it is not an assured interaction. This may or may not happen if the transcription factor is not close enough. Even if it is close enough, there may be some molecules in between due to molecular crowding. So therefore, nothing is assured in biology. Everything is a probability, right? So that is what I'm talking about complexity, and not the number of components, but uncertainty of component interaction. So what's the solution? Practical solution is that we have been good in decomposing the system one part at a time. Now let us compose the system, but this should be a regulated construction, a controlled construction. Biotruth table was proposed back in 2004 when the first event of synthetic biology was organized at MIT. Biotruth table was proposed as a way to go. So for example, if you had to design E. coli, construct 4,000 truth tables and see if we can put the data together in the form of model, or the yeast 6,000 truth tables. But it didn't happen because in reality, we can say logic gates is the way to go. We can make it, sort of an analogy. But in reality, the rocket is never straight. It's not a straight rocket science, not a very deterministic rocket science. There's a lot of probability. So what we end up is making a different version of the reality, what we hope. So synthetic biology is a rational design and controlled construction of biological components leading to applications. People are also calling it biological engineering. In fact, large, now members of the community call it biological engineering rather than synthetic biology, because obviously it is an engineering approach. Just want to highlight for those who are in India, so we do have this special feature that was started a few years back by the Department of Biotechnology and JNU, and now it has evolved into a bigger scale of involvement. So teams from India, they submit these entries every year to the Department of Biotechnology, and we review those applications, and we select 5 teams typically and give them 10 lakh budget each team. And then this team then goes to MIT and presents the work. And so what you see is the picture right now is at MIT. So many teams from India have gone there. They have won silver medal, gold medal, and done phenomenally well. So if you're interested, I think you should have a look at IGEM website and also DBT website. Well, we have a very simple mission. We want to create brand new genes from and brand new proteins from what is called as the dark matter of genome, which does not code for anything, no proteins, no RNA, right? So To provide the proof of the concept, we took intergenic sequences. As you can see, the red colored lines, we took different sequences in different directions, different sizes, overlapping, non-overlapping, and we took all of them, and then we did not know what to take because there is no publication to this. So we took it for the first time. And when we found, so we can, how do you construct a brand new gene from an intergenic sequence is when you take a non-expressing DNA sequence, an intergenic sequence, and you install these signals upstream and downstream. And therefore, by installing the signals upstream and downstream, you can construct the gene. But how do you do it? Where do you do this? So because if you look at the genome sequence level, then this genome sequence is, it's complex to do all the edits there. So we pulled out the information out of the genome and use a certain vector system. So what you see in the next slide here. So we pulled out the information once we bioinformatically clear that this is a brand new sequence which does not have a protein sequence equivalent in nature. We took an intergenic sequence, amplified it, inserted in the vector, expressed it, and the vector itself provided us all the infrastructure for expression. As you can see here, the PBAT topo expression vector by in vitrogen was used, and there we go. So the best moment was found when we expressed these and we could find the proteins on the gel. In the Western blot, we could find the proteins. And this was for the first time that somebody was making proteins from intergenic sequences. So the phenotype of the transform cell, we also checked the structure, but we checked the phenotype, and that was a little more interesting. Because of all the proteins that we made, one protein expression was consistently killing the cell. So we said if that is true, if the killing of the cells is true, then if we switch off the gene expression, can we restore the growth? So then we found that the growth is restored by switching off the gene expression, as you can see in this slide, by switching off the gene expression. So first of all, the cells are getting killed after 2 hours. And when they are killed, you switch off the gene expression and the growth is restored back up again. So thereby proving a linear correlation between the protein expression and the cell growth. The functional study of the lab-made genes, as we call them, is overexpression. If you underexpress them, or if you do not express them, it doesn't matter because nature never recognizes their expression anyway. So then this led to making of an encyclopedia of lab-born genes. When you profile intergenic sequences, introns, pseudogenes, every sequence that you thought is never making proteins, we took those sequences and we said, let's make proteins from them and see what happens. The next few slides will show you the experimental and computational results. So we found a novel antimicrobial peptide coming from the yeast intergenic sequence. And this antimicrobial peptide was resembling an existing peptide, which is defensin molecule. And in terms of structure, they were strikingly similar, almost 98% similar in terms of structure. In terms of the sequence, it was so different. And we found that it is killing all the microbes. Now, where are you getting the sequences from? Those genome sequences which have never expressed, no RNA, no protein. This is the dark matter of genome. Likewise, we found novel anti-malarial peptides. We experimentally determined that these computationally predicted anti-malarial peptides from the yeast genome, when we tested them using the clinical strain of Plasmodium falciparum, we found that in presence of these peptides, 60% of the parasites are unable to enter RBCs. Likewise, we found a novel peptide that kills leishmania, and this was a very interesting story. It has just been, the patent has just been filed. So we said that tRNA never got a chance to express its own protein. It was just doing a kind of a social service for millions of years, but never got a chance. The molecules, but never got a chance to express itself as an mRNA because it is tRNA. It will not express itself. We know that there are steric hindrances. But how about if we allowed the tRNA to make its own peptide? Now we cannot do this in the natural setting. We know this. So we took we bioinformatically, converted the tRNA sequence into an amino acid sequence equivalent using the genetic code table and chemically synthesized tRNA. encoded peptide, as we call it. And we found that at 40 or 30 nanomolar concentration, it is able to completely kill leishmania. And this is amazing effect that we never imagined from a tRNA encoded peptide. And the best part is that even if you increase the concentration of this peptide 10 times or even 20 times, it does not kill the human cells. It selectively kills only the leishmania. So now we are, we have filed it for patent. Likewise, we found some novel anti-breast cancer molecules and some Alzheimer molecules. We're working on that. And this has led to spinning of a company, Foresight Biotech, incubated at JNU. And so we do have some serious discussion with the funding managers to scale up the production of these molecules and a lot more. 2009, the first publication in the series came up in Journal of Biological Engineering, and then I was invited to European Science Commission to deliver a talk on this work. And likewise, there are other organizations they were interested in our work, and so this has led to some global visibility, but a lot more needs to be done. So currently we have close to 500 molecular inventory for pharma and biotech industries. So the best part is that you own the IP of every molecule that you can make. You do not make it if it exists. So you have a lot of bioinformatics filters that prevent you from any such possibilities. So you make only those molecules for which there is no IP, and you make it and you can name it after your name. There are big challenges, of course. Always, the whole idea was full of challenges when it was floated for the first time. But we have come out of that phase, and so we feel that we get a big message from a small atom that be positive, even if you're always surrounded with negative things. So thank you very much for your kind attention, and I'm happy to take questions, if any.
I'm highly thankful to Professor Dr. Pawan Dhar sir for sharing your valuable and insightful lecture on this complex topic, that is reductionism. And I strongly believe that this will definitely help our students as well as other participants to understand the correlation of the biotechnology with this computational biology. So this will definitely help them to further utilize your valuable and insightful lecture in their future research as well as in their career also. Thank you so much, sir, for this insightful lecture, sir. I'm heartily thankful to you, sir.
My pleasure, my pleasure. Anytime, please. So if there are any questions, we can address them, or if we are running out of time, then maybe we can have it later.
Any questions? Thank you, sir. Thank you so much, sir.
Thank you. Thank you. It was nice to have you. Thank you.
Now, I'd like to invite our next speaker for the day, Dr. Zahra Iqbal. Dr. Zahra has done a postdoc from Department of Biochemistry, Chulalongkorn University, Bangkok, Thailand, and a doctorate from National Botanical Research Institute, Lucknow, in plant molecular biology and genetic engineering. Her area of specialization is in plant molecular biology and genetic engineering. She has a number of publications in very reputed journals. We welcome you, ma'am, and request you to please share your thoughts.
Hello, good morning. Am I audible to all of you?
Yes, ma'am, you're audible, ma'am.
Okay, so now let me share my screen with you all. I think my screen is available to all the listeners.
Yes, ma'am.
Okay, so let me start with the presentation here. So as told already, I am Dr. Zahra. I work on camptotranscription factors in Gossypium species, which is also known as cotton. So the topic of today's presentation is genome-wide comparative and evolutionary analysis of Campta family in Gossypium species. So for coming into the introductory part, let me tell you about the Gossypium species, which are cultivated mostly in India, which includes Gossypium hirsutum, Biotrendz, Herbaceum, and Arboreum. So as you can see here, the ploidy levels are also mentioned, which shows that the species have 2N diploid genome with them. So next, I would like to tell everybody the different developmental stages of cotton fiber development. So as you know that the cotton which we use in India is basically the fibers of the cotton. So the analysis part starts from the very beginning, from -1 dpa. Now dpa is day post anthesis. Even when the flowering stage starts, the fiber development starts and it continues up to 50 dpa. So here, as you can see, this is the scanning electron microscopy of different fiber developmental stages. This shows that even before the flowering, the fiber initials are present at -1 dpa. This includes till like second dpa, the fiber initial starts increasing in number and in length. And as the dpa increases, the fiber initials also increases in length and they start budding out. With 19 and 25 dpa, we have the whole mature fiber initials, which is finally woven into cloth or whatever purpose the cotton is used for. So next, this is the 50 dpa cotton fiber that is used basically for the textile industry. So next, coming on to the transcription factor in for which I'm doing the study is it's CAMTA, calmodulin binding transcription activator. So let me tell you a brief history of how CAMTA works. So whenever there is any biotic or abiotic stress or a developmental signal perceived by a plant cell, there is a rapid and transient increase in calcium ions. So this increase in cytosolic calcium ions is sensed by a number of calcium sensor molecules such as calmodulin, and then this calmodulin in a calcium dependent manner regulates several transcription factors resulting in gene expression and drawing an appropriate response by the plant. So very briefly coming on to the CAMTAs, as previously told in the last slide, the biotic and abiotic or growth signal results in calcium, which activates the calmodulin molecule, and then this calmodulin molecule binds to the calmodulin binding domain of the CAMTA transcription factor. The most important factor involved with this CAMTA transcription factor is the domains. This is the CG1 domain, which is implicatory to be had by this transcription factor. CG1 domain is a unique DNA binding domain, and for any transcription factor to be characterized as CAMTA, this CG domain, which is CG-CG or CG-TG, should be present. The other is the transcriptional activation domain, which is responsible for the transcription. The TIG domain, which is responsible for non-specific DNA interactions. Enchirin, which is responsible for protein-protein interaction. And of course, the calmodulin binding domain, which results in the calcium calmodulin binding to the transcription factor and resulting into the eventual activation of this transcription factor, withdrawing the response. And since CAMTA is a transcription factor, it is generally localized into the nucleus. So for cotton, I took 3 species, that is Gossypium arboreum, Ramundi, and Herbaceum for the study, and found that in Gossypium arboreum, 6 CAMTAs were present, Gossypium ramundi 7, and Gossypium herbaceum 9 CAMTAs were present. Along with them, their cotton IDs and chromosomal locations were also studied. And all of them were found to be localized into the nucleus. Next, as told in the previous slide, that CAMTA have unique domains. These were searched for the domain organization for the presence of CG1, TIG, and Enchirin, and all the Gossypium CAMTAs were found to contain the obligatory CAMTA binding domains. So for CAMTA, calmodulin is very necessary molecule, and when the conservativeness of calmodulin molecule was seen in different CAMTAs in comparison to the Arabidopsis CAMTA, they were found that the calmodulin domain is highly conserved in cotton CAMTAs. Even when the peptide amphipathic molecule was drawn, it was seen that the calmodulin has an amphipathic nature in Gossypium species, which corroborates its role with the Arabidopsis campta. This is the phylogenetic relationship of the Gossypium camptas with different plants, and as you can see that all the camptas are grouped together in group 1A along with the Arabidopsis, which shows that they are having a higher phylogenetic relationship with the cotton with the Arabidopsis campta. And one more interesting finding was that the Gossypium is highly similar to Threobrama cocoa, which is also shown by this phylogenetic analysis. And both these species, that is cotton and Threobrama, belong to Malvaceae, and so they are grouped together. So this is the phylogenetic relationship of different cotton camptas among themselves, showing the exon-intron organization and the motifs present in them. All the cotton camptas were found to have similar kind of exon-intron and motif organization amongst themselves. So next, we took like 6 Gossypium herbaceum camptas, candidate Gossypium herbaceum camptas, and saw their expression profile in different fiber developmental stages. We had the in-house transcriptome data available with us. So the stages of fiber development which we took was 0, 6, 9, 12, 19, and 25 GPa. It was found that GH campta 7A was highly expressed in the later, initial stages of fiber development, whereas Gossypium campta 2A.2 was highly expressed in the later stages of fiber development. The other camptas, that is 3D.1, 4D, and 5D.1, were expressed to a significantly lower levels in different developmental stages of fiber. So this is the correlation expression network of the two highly expressed campta. Firstly, we will be talking about campta 2A.2. These were the positively co-expressing genes which were related to 2A.2 campta, and this were the negatively co-expressed genes which were correlated to campta 2A.2. The regression were taken as highly significant, that was 0.95. So these positively co-expressed genes were further plotted for the expression profiles at different DPA, and it was shown that at all the DPAs, these positively co-expressed genes had a similar kind of expression profile. For the negatively co-expressed genes, the negatively co-expressed genes were highly expressed at 5 and 10 DPA in comparison to the positively expressed genes. When these genes were analyzed for their pathways, in which pathways they were involved, so it was found that these genes were mainly involved in developmental or stress-related pathways. Moreover, the 1000 base pair upstream region of these positively or negatively co-expressed genes were also searched for the presence of campta recognition motif, as told before, CGCG or CGTG, and it was found that the positively and the negatively co-expressed genes had a higher frequency of the campta recognition motif in comparison to the random set. The similar study was also conducted with campta 7A, in which the positively co-expressed genes were almost similarly expressed in different fiber developmental stages, but the negatively co-expressed genes in comparison to the 2A.2, they were highly expressed in the 10th DPA stages. Also, these genes were also reported to play a role in developmental and stress biology. Again, the positively and negatively co-expressed gene had a higher frequency of campta recognition motif in their promoter region in comparison to the random set of genes. To further corroborate our study, what we did was we plotted the correlation expression coefficient with different fiber-related parameters, which included fiber strength, fiber length, micronair, uniformity ratio, ball number, and ball weight. Now, ball is a structure from which the fiber initiates, like you take out the fiber from a ball when it erupts. So for the two highly expressing camptors, that is 2A and 7A, the regression was found to be significant in terms of fiber strength, indicating that probably these two camptors are playing good role in imparting fiber strength in Gossypium species. The other camptor, that is the lower expressing camptor 3D.1, had a significantly lower regression coefficient with different fiber related traits. So this campta was seen as not a candidate gene. However, campta 2A.2 and 7A were seen as a candidate gene in fiber strength. So further, with QRT-PCR at different fiber developmental stages of cotton campta, the expression profile of 2A.2 and 7A and the lowest expressing campta 3D.1 was observed, and it was seen that campta 2A.2 was highly expressed in the later stages of fiber development, and campta 7A was expressed in the initial stages of fiber development, which was also corroborating our in-house transcriptome data. However, the least expressing cotton campta did not expressed highly in any of the tissues. However, it was significantly declined as the fiber developmental process progressed. So very quickly coming on to the conclusion, the study comprehended the underpinning molecular, biochemical, and physiological aspects of campta in developmental regulation, perhaps proposing the role of campta in fiber development. And the study also provided an insight of campta by modulation of different hormonal and signaling pathways. I thank the whole team of NIT for giving me this opportunity to present. Thanks a lot.
I'm highly thankful to Dr. Zehra Iqbal, ma'am, for her valuable lecture in the field of genetic engineering and plant biotechnology. And I firmly believe that this lecture would definitely help our young biotechnologists as well as other participants in enhancing their research and academic capabilities, especially in the field of biotechnology. And also it will definitely help them to explore more in this area. Thank you so much, ma'am, for your insightful lecture and your presence in our international conference. Thank you so much.
Thank you so much. I'm happy to take any questions if there are any.
I would like to ask audience if they have any query, they can type in the comment box and we'll get the issue resolved. Thank you so much.
Thank you so much. Thank you so much, all the team.
Now, I'd like to introduce our next speaker for the day, Dr. Nour. Dr. Nour is a professor in the field of environmental and petroleum biotechnology and head manager of Petroleum Biotechnology Lab, Egyptian Petroleum Research Institute, Cairo, Egypt. Dr. Nour is an author for 5 books in the field of biofuel and petroleum refining and biotechnology, and about 120 research papers in the field of oil pollution, bioremediation, biofuels, microbial corrosion, green chemistry, wastewater treatment, and nanobiotechnology and its application in petroleum industry and biofuels. Dr. Noor is also an editor in 16 international journals and reviewer in 18 international journals, supervised 22 MSc and PhD theses in the field of biofuels, macro fouling, bioremediation, wastewater treatment. Dr. Noor participated in 26 international workshop and training courses and 37 international conferences. Dr. Noor is a member in many international associations concerned with environmental health and science. She was also a lecturer and supervisor for undergraduate research project at the British University in Egypt, BUE, and Faculty of Chemical Engineering, Cairo University, Egypt, and also taught Environmental Biotechnology course for postdoctorates at Faculty of Science, Montefiore University, Egypt. Her biography is recorded in Who's Who in the Science and Engineering 9th edition 2006-7. We welcome you, ma'am, and invite you to please enlighten us with your thoughts.
Hello, can you hear my voice?
Yes, ma'am.
Okay, can you see my presentation now?
Ma'am, not yet.
I have clicked on share, so what else I can do?
Ma'am, we can only see the folders.
The folders?
Yes, ma'am. We can only see the folders.
I will try to share it again.
Okay.
Now, is it okay?
No. Yes, ma'am. It is visible now.
Okay, thank you. It's okay now?
Please switch the slideshow. Ma'am, please switch on the slideshow mode.
So you can see now? Can you see the presentation?
Yes, ma'am. We can see the presentation. Please switch on the slideshow mode, ma'am.
I don't know. If you can see, okay?
Okay, ma'am. Okay, ma'am. Fine.
Okay. Good morning. I'm Nour, Professor of Nour Shafik Emam from Egypt. Thank you for introducing me, and thank you for inviting me to give this talk with your colleagues in India. I am here from Egypt now. It's about 7:30 a.m. in the morning. My talk will be about sustainability and how we can apply nanobiotechnology for reaching to green and sustainable environment. I am Professor of Petroleum and Environmental Biotechnology. Now I am the head manager of Petroleum Biotechnology Lab in Egypt, in Egyptian Petroleum Research Institute, vice head of Center of Excellence in one of big universities in Egypt, which is Modern Sciences and Arts University. I'm also the coordinator of Nanobiotechnology program for Faculty of Nanotechnology for postgraduate studies in Cairo, Egypt, vice coordinator of the Scientific Research Committee, National Council for Women in Egypt. My talk will talk as an introductory part to what's about biotechnology and its applications. As you can see, biotechnology has different applications in industry, like in medicine, in agriculture, in non-food industries like that of biodegradable plastics or bioplastics, biocatalysis, biofuels, which is alternative or complementary to different kinds of petrofuels, and also for environmental uses, for removing the reclamation of environmental waste or for removing of waste, do some kind of waste management. Other applications of biotechnology, the use of organisms to manufacturing organic products like that of beer and milk products, in mining industry for, like, for example, bioleaching, for waste recycling like that of waste management, like that of composting of lignocellulosic waste, for example, or also in the cleanup of contaminated sites like that of bioremediation or biosorption in water, or also we can use it for desalination. Oil and gas industry, like that of the upgrading of petroleum and its fraction, like that of removal of sulfur or nitrogen, or by upgrading of heavy crude oil to lighter crude oil, and also for microbial enhanced oil recovery. And now to produce biological weapons like that of all we're suffering all over the world now. So biotechnology has different colors, and this briefly give it to you the classification of biotechnology by colors. The gold going to bioinformatics and nanobiotechnology applications in general. The red is the application of biotechnology in medicine and human health. White one is the application of biotechnology in biofuels, for example, in industrial, like that of biofuels, biopolymer, and biocatalysis. Yellow is the application of biotechnology in the food industry. Come the gray, which is the bioapplication or biotechnology application in environmental applications in general. Green, which is the application of biotechnology in agriculture field, like that of biofertilizers and biopesticides. The blue is the application of biotechnology in marine environment and the use of the marine resources in anything like that of application of microalgae or macroalgae. Brown, which is the application of biotechnology in desert and dry region. Violet or purple is concerning with the patents, laws, ethical and philosophical issues related to biotechnology. Excuse me, you cannot see my presentation? Hello? Hello?
Ma'am, actually slides are not moving. If
Excuse me?
you can allow me, can I share your slide, ma'am?
Okay.
Can I share your presentation?
Okay.
If possible, can you tell me, ma'am, on which slide currently you are having, ma'am? You're running on?
I can, if you would like me to start, okay.
No, I can start, ma'am. I can share.
Okay. I mean, that's the third slide.
Okay, fine, ma'am. I'm just sharing it. Just... Ma'am, is it okay? Is this the slide, current slide?
Excuse me?
Ma'am, is it your current slide?
I cannot see the slide now. Please try to give me the control. Excuse me?
Yes, ma'am.
Hello?
I have given you the control, ma'am. If you can see, I have given you the control.
I can, yes, okay. Can you, I cannot use it now. I'm sorry. I cannot. Can you please make the share with the third slide?
Ma'am, is it okay?
Yes. Can you enlarge it, or you will keep it like this?
No, ma'am. I'll enlarge it. Is it okay, ma'am?
Yes.
Yeah. Ma'am, if you allow me, I can move the slides as per your lecture, ma'am.
Okay. Okay. No problem. But can you please open it now? This is the fourth slide. You please go back to the third one.
Is it okay, ma'am?
Okay, but this is not full screen. Is it okay for if it's not full screen?
Just wait for a moment, ma'am. Let me set it.
I cannot see the presentation. Hello?
Yes, ma'am. Ma'am, just give me one moment, ma'am. I'm just trying to set it, ma'am, as per your. No. Ma'am, is it okay? Hello?
Hello?
Yeah, ma'am, is it okay, ma'am?
It's not a slide. It's not a slideshow. It's not full screen. Hello?
Ma'am, if possible, can you continue with this, ma'am, slideshow? Because it is visible from our side, ma'am, if you permit us.
You can see it as a whole screen? Hello?
Yes, ma'am.
I cannot see the presentation now.
Ma'am, is it now visible? We have done the
No.
slideshow.
Yes, this is. Yes, okay.
Fine, ma'am.
Can
Start.
Can you give me the control to return to the previous slide, or you will do it?
Ma'am, I'll move the slides as per your need. Means as per your lecture, I'll move the slide, ma'am, as you suggest.
Okay, so we can restart from the beginning, from the first slide, or what?
As you wish, ma'am, because the introduction was fair enough. We have gone through that. So you can start from here also, ma'am.
So could you please go to the third slide, please? No, have you seen this? Okay, we can go to the third slide. So we are reaching now. This is, as I have spoken from a while, about the colors of biotechnology. As you can see, we have about the gold one, which is related to nanobiotechnology, the red, which is related to the medicine and the human health, the white, which is related to the industrial biotechnology, like that, biopolymers, biocatalysis, and biofuels, the yellow, which is related to the food industry and nutritional sciences, the gray one, which is related to the environmental application, the green, which is related to the application of biotechnology in agriculture field, like that of pesticides and biofertilizers, and the blue one, which is related to the marine, and the brown, which is related to the deserts and the dry region, the violet, which is related to patents, laws, ethical issues, and philosophical issues related to biotechnology, and the bad one, which is the dark, which is related to the bioterrorism and biological weapons. Now, will concentrate on the application of nanobiotechnology in general and how we can use all our resources and the waste resources to valorize it for valuable nanomaterials and nano oxides and nanoparticles to different application in the field of biotechnology. So waste and biomass valorization to produce nanomaterials. So can you go now, please? Next slide, please. Now, as we can see, we can achieve the 17 goals of sustainable development by application of nanobiotechnology, because when we valorize waste or biomass into valuable nanoparticles, and with the application of it to reclaim different types of pollution, like that of underneath water or the on land, so you can save and achieve two of the main goals, which is save life underneath water and save life on land. Also, when you use waste management or use the waste resources, you can save food and you can protect your environment. So you can protect your land from deforestation. So you will, and you can apply it for the production of biofertilizers and biopesticides. So you will have a good source of food all of the time. Also, you can overcome the problem of climate change by the application of nanobiotechnology, and I will show you how we can do this. And also, you can secure yourself by having new sources and resources for clean energy, and also for clean air, and also for society, and overcoming the poverty by having some sources of new jobs for unemployment and solve the unemployment problem. So when you have a good environment, you will have a secured society and a good economy. Now, by application of nanobiotechnology, you can provide the 3 pillars of sustainability and the 17 goals of sustainable development. Okay. Next slide, please. So when we talk about sustainable nanotechnology, it is a very locally available, a local availability. It is a cost-effective process. It is socially acceptable. It is eco-friendly. Application of nanotechnology at the green nanotechnology can be applied in wastewater treatment and water desalination, biotechnology and agriculture, medicine and health, food preservation, in energy and the environment. So you can do or achieve the 3 pillars of sustainability. Okay. Okay. Because our aim is health and safe environment, so I will show you some different applications of nanobiotechnology we have achieved. This is part of our publications that we have already published in different journals, international journals, and the publication on books and also patents. Can you please go on? First of all, I would like to talk about the problem and how we can solve it. As we can see, with the increase of the population all over the world, there will is a great increase in the demand of petroleum. By year 4, it will increase in about 40% by the year 2025. This comes with the depletion of the fossil fuels or non-renewable fuels in general, especially the petroleum with high quality, low sulfur content. So all of this will lead to the increase of global warming, and also because of the fluctuation of crude oil prices, we have facing a lot of economical problem all over the world. By the emissions of the bad or the non-incomplete combustion of petrofuels, we have the increase in particulate matters and the greenhouse gas emissions, which leads to, and also SOx and NOx, which leads to the acid rains and to the problem of climate change. Okay. This is when we can, I will show you something. We have two types of crude oil, heavy crude oil and light crude oil. The heavy crude oil is the asphaltenic crude oil, which has high concentration of asphaltenes. So this is the main structure of this asphaltene. If you would like to bio-upgrade the crude oil and from increasing its quality from high crude oil or the heavy crude oil to the lighter crude oil, you should de-asphalt this product. You can de-asphalt it by chemical process, which is very expensive, but you can apply this by applying biotechnology and nanobiotechnology to increase the rate of bio-upgrading by degrading or the heavy high molecular weight asphaltenes to the lighter fraction, which is the maltene. Okay. These are one of the most problem when oil spills occurs. The persistence of these biomarkers or persistent biomarkers in the environment for a long period of time has a very bad negative impact on the ecosystem. Okay. Effect of oil pollution, as we can see, it affects tourism, destroys shorelines, it affects the life underneath water, the fish population, the seabirds, the coral reefs for a very long period of time if oil spills occurs. So if as long as oil spill occurs, you should quickly reclimate it from the environment. You can do this by physical methods or chemical methods, by using dispersants or by using booms. But upon the application of bioremediation, you assure, or the nanobiotechnology, you assure that the complete reclamation of the pollution occurs. Excuse me. Oil pollution not only affects the water, but it also affects the on land. For example, because usually the high heavy metal content is concomitant with the oil spill, which leads to reduce the soil microbial activities and the biomass. When the plant bioaccumulate these heavy metals or some of the oil component like that of the aromatics, this also negatively impacted the plant. And when we animals eat this, it is negatively impacted or affected by these pollutants and its organs accumulate these heavy metals and polyaromatic hydrocarbon. So indirectly, human can be affected by the oil spill from the land, on land or in water. Okay. These are examples. As we can see, these are some samples from fish that were collected from water that have been subjected for oil spill. As you can see, it mainly has these are the 16 polyaromatic hydrocarbon represented by the U.S. Environmental Protection Agency as 16 purity pollutants. As you can see, it mainly has high concentration in its organ from the 6-member polyaromatic benzopyrene, which is toxic, mutagenic, neurotoxic, genotoxic, and carcinogenic. Okay. So you now can see the problem of having any oil spill. It persists for a long period of time and accumulated in the organ of fish and plants, which we are eating it through animals or vegetables or plants. Another problem is the main problem, which is the biocorrosion. Biocorrosion is mainly formed from one of microorganisms like that sulfate-reducing bacteria. The sulfate-reducing bacteria can live on over a wide versatility of pH, temperature, and salinity. It is mainly living in under anaerobic condition. First, by the formation of the aerobic biofilm of aerobic microorganisms, and when the media in these microorganisms attach it to the metal or the surfaces or solid surfaces, now the media underneath it by producing of exopolymeric substances, which is mainly of polysaccharides, make it nearly anaerobic area. It's ready for the cross of the sulfate reducing bacteria, which is very corrosive, producing the hydrogen sulfide, which is a very corrosive environment and causes the pitting corrosion and the sudden leakages of the pipelines, which leads to very bad impact in the environment by the leakage of petroleum and other wastewater. So we should overcome this problem and killing the sulfate reducing bacteria by a safe nanobiocides. Okay. Another problem to be solved is the high concentration of the solid biomass all over the world. Egypt suffers from approximately 60 million tons of biomass of organic wastes. Comes nearly 60% of which was organic waste. This solid waste can be used for the production of nanomaterials, and for example, for the production of biofuels, another example for the production of biogas and valuable products that can solve the problem in a very cost-effective way of waste management. Okay. Now, these are the three types of waste recycling. This can be by recycling, downcycling, or upcycling. Recycling, which is from plastics to plastics, as you can see, by reduce, reuse, recycle. These are recycling. What about downcycling? When we use, for example, white paper to prepare cardboard, this is downcycling because we can reuse it, but usually as to produce a lower value product. However, this comes the upcycling, which is using the or low cost products to produce a valuable value added product. This is upcycling. So by waste management in this area, so you can have a four-fact solution for economy, energy, environmental, and waste management. Okay. Okay. Another way, which is biodiesel. Why we go to the biofuels in general, and for example, biodiesel and bioethanol, because, for example, biodiesel is a good alternative to the petrosolar or petrodiesel. Is the solar. It is non-carcinogenic. It has no aromaticity. It cannot emit greenhouse gas emissions in general. It's nearly a closed cycle of carbon dioxide. It's about 78 reduction when we apply or use biodiesel. It's 78 production of carbon dioxide emitted. So we can overcome the problem of climate change by the application of biofuels like that of biodiesel. Biodiesel has nearly the same qualification like that of petrodiesel, so it can be used in the diesel engine without any modification. Okay. In Egypt, we have, as I said before, waste biomass, for example, of 60 million tons per year. Most of it can be used for the production of biofuels in general. Even the municipal waste, we can use municipal waste to produce biogas in general, or you can also use for the production of microalgae, which can be used for the production of biodiesel and also biogas. Okay. Now, the main problem we're facing all over the world is that they usually produce the biofuels in general from first generation, second generation, and third generation. The first generation is the production of biofuels from food, which food crops like that of corn. And this is very bad. And then the second generation, which is using, for example, non-edible oils and waste cooking oil. And the third generation, which is also recommendable, the second and third one is recommendable. The third generation is from using algae. The main problem is that up to now, they depend on the first generation. And as you can see, this chart summarizes the problem as 8 bushels of corn produce approximately 22 gallons of ethanol of fuel per year, which is enough to feed a human for a whole year. 1 bushel is equivalent to approximately 25.4 kilogram of corn. Now you can see the problem of food versus fuel. Okay. The major drawbacks, as we should solve by application of nanobiotechnology in the preparation of the biodiesel. The main process is depending on the transesterification. Even when you use the second generation or the third from algae or from the waste cooking oil and non-edible oil, the problems is from the homogeneous catalyst that can be applied, which is acidic or basic catalyst. It is non-reusable. It's very costly or expensive because it can be used for only once. It's also you can consume a lot and a huge amount of water in the neutralization or the washing step. It's also when using acidic catalyst, it is very corrosive and needs high temperature and pressures, which adds to the expense of the process. And so when we apply, you can solve this by using basic catalyst, but you have the main problem, which is washing step, which consumes a huge amount of water, and the catalyst can be used for only once. Applying, okay, can you go to the second slide? Applying heterogeneous catalyst, okay. Second slide, please. Applying heterogeneous solid catalysts can solve most of this problem because heterogeneous catalyst can be used for different times or at least 5 times without losing its activity. You can prepare this catalyst from cheap sources, which is readily abundant like that of the waste, as I will explain later. And this is also in the nano range, so you can apply nanobiocatalyst for the production of biodiesel, which will decrease when applying waste cooking oil and the catalyst prepared from waste sources or readily available waste sources. This will decrease the price of the biodiesel by reaching to approximately 9% of its cost. Okay. As I have said before, why biocatalyst from waste cooking oil? Because it will decrease the price. It's readily available source. You will overcome the problem of food versus fuel. Okay. Why bioethanol? Bioethanol is another source of biofuel, which you can also apply nanobiocatalyst in its different process, from the fermentation process in the pre-treatment process to produce bioethanol. Mainly, the main important advantage of bioethanol is the decrease of the emission of carbon dioxide by approximately 70%. It can be used as a complementary to the petrol or the gasoline. Okay. Disadvantage of bioethanol is its calorific value is nearly 30% lower than that of the gasoline. And also the main disadvantage of bioethanol is that it's up to now the main source of production of bioethanol is the food sources. Okay. When we apply the third generation, which is mainly from the the second generation, which is mainly from the lignocellulosic waste, or even the third generation, which is mainly from algae, the main problem is comes from two parts. First is the pre-treatment to make the celluloses and hemicellulose, which is the sugars available for the enzymes to produce the sugars for fermentation. So to overcome this problem, you should have a good isolate or good enzymes for the pre-treatment process to produce the celluloses and hemicelluloses, and also you should have a good isolate for the fermentation of hexoses and pentoses. And also you can apply nanobiotechnology to accelerate the rate of the pre-treatment process and the fermentation process. Okay. Now I will show you some examples of our publication for reaching sustainable nanobiotechnology. These are the example. When we apply and produce bioethanol, we have in the fermentation process a huge amount of biomass as a spent waste. This biomass can be used for biosorption and pre-treatment, a biotreatment of water polluted with dyes like the basic blue dyes. And then by applying titania as a hydrosol, we can regenerate this biosorbent, and it can be used at least for 3 times without losing its activity. Another example, please. Next slide. Same, we have used this biomass spent waste after the production of bioethanol for the bioabsorption of another type of dye and then regenerate it by another type of nanocatalyst, which is also prepared green by green synthesis, like that titania is anatase and rutile, and we have applied this for photo degradation of the dyes to reaching to complete mineralization of the dye for the recycling of the water and also for the recycling and pre-preparation or reuse of the biocatalyst or the biosorbent, excuse me. Okay. We have also managed to increase the rate of bioabsorption by the mixing of the spent waste of the biomass produced from bioethanol fermentation process with the polyaniline nanocatalyst as you can nanosorbent. As you can see, it increases the bioabsorption rate and the removal of the sulfonated acid red dyes. Okay. Another example is the animal bone. By simple calcination, we have managed to produce the fluoroapatite and the hydroxyapatite nanocatalyst, which has good viscosity, good specific surface area, poor volume, and poor diameter. And okay. This catalyst has good photo degradation capabilities and can degrade water and reclaim water from coming out from the petroleum refinery, which has high concentration of phenols and chlorophenols. Okay. We have also managed to use this catalyst, biocatalyst or nanobiocatalyst prepared from waste, to use it as a biocatalyst in the transesterification of waste cooking oil, and we have managed to produce biodiesel, the same, which is more beneficial because it decreased the cost of the process. We have managed to produce approximately 96% transesterification of waste cooking oil compared with the commercially Novozyme, which is expensive enzymes used for the production of biodiesel. And the spent waste of biocatalyst, which is fluoroapatite after the transesterification process, for reaching to the point of zero waste, we have managed to produce it as a photocatalyst for the photo degradation of nitrophenol. So we can reclaim water and also produce biodiesel from this animal bone nanocatalyst. Okay. We have also managed from the waste like that of watermelon peels to produce another types of nanocatalyst, which is zinc oxide, which has a good photo degradation capabilities for reclamation of water, like that as we have managed to photo degrade malachite green. Okay. Applying this zinc oxide, green synthesized zinc oxide, and the animal bone nanocatalyst, we have managed to use it as a biocide for the sulfate reducing bacteria, which is the main cause of biocorrosion. Okay. We have managed also from the water extract of some peels like that of outer brown skin onion or outer skin of garlic or orange peels, mandarin peels, and the wastewater from henna leaves and lupine, better water lupine. This extract, water extract, is also in the as like that of nano emulsion, have been used for as a corrosion inhibitor for the chemical corrosion produced from in the acidic media. Okay. This scanning electron microscopy show that the nano Dept of this extract on the, as you can see from the right hand micrograph, the smooth surface free of any corrosion, and the left hand is we have the spitting corrosion caused by the acid media. Applying these nano emulsions, it has a good corrosion, act as a good corrosion inhibitor. It's safe, green, and eco-friendly and cost-effective. Also, this extract and nano emulsion have been used as a biocide against different types of sulfate-reducing bacteria, halophilic and halophilic 1 and non-halotolerant. And also, it acts as a good biocide for mixed culture collected from different Egyptian oil field. It also biocide can be act as a biocide for macrofoulants, not only microfoulants. And it has the main important thing, it's cost, and it is cost effective and eco-friendly. It has less toxic effect to non-target sea organisms like that, isopods, amphipods, and decopods. Okay. Also, we have managed from this extract a nanoemulsion to green senses, for the green senses of hematite nanoparticles. This hematite nanoparticles has a good band gap, which is can be used for as a photodegrader. Okay, go to the next slide, please. As a photodegrader, it has a good photocatalyst as a good photocatalyst for different pollutants, chlorophenols, and different types of dyes. Okay. We have also managed by the water extract of orange peels to produce silver nanoparticles. The silver nanoparticles has a good biocidal effectivity against and bactericidal activity against pathogenic microorganisms, and is not only bacteria but yeast and fungi. And it is cost effective because it costs approximately $81 per 10 gram of silver nanoparticles, where compared to that of the global cost of the chemical synthesized silver nanoparticles, which is $250 per 10 grams. So we reached to saving for approximately 68% saving by the green synthesis of silver nanoparticle using the waste peels of orange. Okay. We have also managed by the using mandarin peels to produce silver nanoparticle. The cost was approximately $0.76 per gram of green synthesized silver nanoparticle. And the waste after the spent waste after the production of silver nanoparticle have been used for the production of the valuable activated carbon, which is also in the nanoscale. This activated carbon has different application, like it can also be used as adsorbent. It can be also used as a mobilizing matrix for bacteria for different application. The activated carbon also can be used as a in the preparation of biodiesel as a catalyst. Okay. We have also managed from this green synthesized sulfur to be act as a biocide for the biomass from sewage water. So you can reclimate the pathogenic water to be infected water to be reused for agriculture. Okay. And also we have managed to prepare biologically using fungus, so silver nanoparticles. And as we can see, okay, these silver nanoparticles have been used as a biocide, and the green synthesized or biosynthesized silver nanoparticles as a biocide for the sulfate reducing bacteria. As you can see, the intact SRP, the cells is free and is alive, and the cell wall is not intact. After the application of silver nanoparticle, as you can see, cell leakages occurred, and the death of and the flagella of the sulfate reducing bacteria is broken, and so the death occurred. And the biocide silver nanoparticle, we have approved it as a good has a good biocidal activity against the sulfate reducing bacteria. Okay. We have also managed by biological methods for using application of aspergillus as a fungi. Its extract, a water extract of its mycelia have been used for the preparation of cobalt oxide with good super paramagnetic ferromagnetic properties. This nano oxide have many applications in industrial field, and also has a good biocidal activity against pathogenic microorganisms. Okay. Another problem, as I have told you before, the bioupgrading of crude oil. Now we are trying to solve this problem by the application of nanobiocatalyst. Okay. Next slide, please. We have managed to prepare ferromagnetic nanoparticles. The ferromagnetic nanoparticles when we decorate the microorganism, different microorganisms, the gram-positive, gram-negative microorganisms in general, it has different application. Because upon applying this coating, by coating this microorganism, you can solve the problem of separation of microorganisms from the bioreactors so easily, because one of the main costs coming from the bioprocesses in general in any industrial field is how you can reuse the microorganism without losing its activity. How can you protect it from contamination? How can you increase the lifetime of the biocatalyst? How can reuse it? How can you separate it without in a very cost-effective process? Because one of the major problems is the separation by, for example, centrifugation. It's very cost process and also expensive and waste a lot of energy and money. So by applying magnetic nanoparticle, you can solve all of these problems. As you can see, this magnetic particles has a good base, specific surface area, poor volume and poor diameter. It can absorb and coating the microorganisms without affecting the enzymatic activity of the microorganism, and it's very safe to the microorganism. And also it has good absorption capacity to some compound like that of sulfur compound, and also the pyrene, and also the carbazole as an example of the nitrogenous compound. So next slide, please. Next slide, please. Hello. Excuse me.
Yes,
go to the next. No, you have jumped and the slide before, please.
Okay.
Go up, please. Another slide up.
Just wait for a moment, ma'am.
Okay. The slide 4, please. Yeah, thank you. This is an example when you apply, for example, the magnetized microorganism, or by coating the bacillus, for example, by the magnetic nanoparticles. As you can see, it's well absorbed on the surface of the microbial cell. The magnetic nanoparticle has no toxic effect to the microorganism. The coated cells show higher biodesulfurization rate, higher biodenitrogenation rate, higher biodegradation rate than the free cells. It can reach to complete removal, for example, compared with approximately double the rate of biodesulfurization, double the rate of biodegradation, double the rate of biodenitrogenation. And also, as it can be reused for at least 4 successive times without losing its activity. It's also tolerable to the toxic intermediates produced during the bioprocesses. It can be stored for at least 1 month under 4 degrees without losing its activity. It can be used for a long period of time, so you have overcome the problem of the short lifetime of the catalyst. It has the main advantage of the magnetic separation, which would resolve many operational problems. Okay. But when we apply this and as a case study, we have tried to do this apply this on diesel oil for desulfurization. The main cost for the process comes from the preparation of the magnetic nanoparticle, chemically prepared magnetic nanoparticles. And also the main cost comes from the preparation of the biomass itself. The media component is expensive. Now, by applying the green synthesis or the nanobiotechnology, by applying the green synthesis of the magnetite, we have overcome this problem. The cost of the process of the preparation decreased by approximately to the tenth of this value. And also we have managed to the green synthesis, we have overcome the problem by of the cost of the preparation of the media of the biomass by applying the agro-industrial waste like that of molasses and corn steep liquor for the preparation of the biomass. Okay. Okay, I have already talked about this. Now, to catch the wave of biofuel, as I have told you before, we can prepare bioethanol as example of biofuels and biodiesel as example of the biofuels also. We can apply magnetite, the green synthesized magnetite, as a catalyst for the transesterification process. We can also apply the magnetic nanoparticles to increase the rate of the cellulase production and for the pre-treatment process, the biomass and waste management, for the sugar production and for the fermentation process. Okay. This is another type. We have managed to prepare from eggshells, mollusk shells, and animal bones nanocatalyst like that of calcium oxide and fluoroapatite and hydroxyapatite. Okay, go to the next slide. to produce biodiesel. Okay. This is the process, summarizes the process. It's simple calcination process. Just you grind the waste, and then by simple calcination, you can produce the catalyst in a nanoscale. Okay. And by comparing its activity with the commercially available chemically prepared calcium oxide and the expensive Novozyme, as you can see, the range of the production of biodiesel using the nanobio-catalyst prepared from waste can range from 90 to 93%, which is higher than that of Novozyme and within the same range of like that of the produced biodiesel using the chemical calcium oxide. The main advantages is that the cost decreased by of the production of biodiesel using this nanobio-catalyst, reaching to approximately $2.5 per gallon biodiesel. Okay. As we can see, by applying this biodiesel and applying using this nanobio-catalyst prepared from waste products or waste biomass, the greenhouse gas emission decreased in general. Okay. We have also managed by silica produced or extracted from rice straw to be used as immobilizing matrix for the enzymes like that of gram-positive Bacillus stratoferricus. This we have managed to produce lipase enzymes, which can be used for by the immobilizing it by using the silica extracted from rice straw to produce biodiesel as used in the transesterification process. As compared with the commercially available Novozyme, the Novozyme have been used for only once, but our new immobilized catalyst can be used for at least 6 times without losing its activity. Okay. Another way for the application also of nanocatalyst is when you produce by, for example, you can solve the problem of wastewater treatment and also produce the biomass of algae, which can be used for the production of lipids, which can be used for the production of biodiesel. As you can see, we have used the tetrasalamus for the phycodegradation of one of the examples of pyrene, one of the pollutants coming out from the petroleum hydrocarbons, this carcinogenic high molecular weight polyaromatic hydrocarbon. As you can see, this algae, after the rate of the biodegradation can be increased by the application also of magnetite nanocatalyst or other nanobio-catalyst, and also this lipid can be used for the produced from this biomass can be used for the production of biodiesel, and the waste biomass after the production of biodiesel can be used for the production of biogas by anaerobic fermentation. Okay. So you have reclaimed water, you have done wastewater treatment and produces biofuel. Another way for also the application of nanobio-catalyst, also by applying spirulina, you can produce lipids, you can produce different types of proteins, carbohydrates, and nucleic acids, and also different types of natural dyes. Okay. This is another example for the fermentation process. If you have lignocellulosic waste like bagasse, you can produce bioethanol, and the spent waste can be used for the water bioabsorption or by absorbing the oil spill, like that oil spill of kerosene or diesel, and the solid waste after the bioabsorption process of the biotreatment of water can be used for the production of solid biofuel with good calorific value. So you have reached to zero waste and another type of source of fuel. And also the spent waste like that can be used for the preparation of after fermentation. The wastewater after fermentation can be used also for the preparation of or the green synthesis of nanocatalysts with different applications. Okay. This is another type or source of biofuel, the macroalgae, which is very abundant on all of our seas and oceans. You can use this macroalgae for the preparation of bioethanol as a third generation biofuel. Okay. This diagram can summarize to you how you can produce different types of nanomaterials with different application from the one source of lignocellulosic biomass, and also by producing different types of biofuels to reach for sustainability and complete life cycle assessment. From lignocellulosic waste biomass, we can, by cost-effective hydrothermal pretreatment, you can prepare the celluloses and hemicelluloses. The lignin produced from this step can be used for the green synthesis of metal and metal oxide nanoparticles with different application. The cellulose can be used also for solid state saccharification or enzymatic hydrolysis, can produce enzymes. Also can produce the sugars which can be fermented to different biorefineries like that of bioethanol, bioethanols, and bioacids. The yeast coming out from the process of fermentation can be used as a single cell protein, and also this with the spent waste biomass can be used for the biotreatment of water, and after that the waste with the pollutant can be used for as a refuse derived fuel, as a solid biofuel. The metal oxide and the activated carbon produced from this waste biomass can be used for as a catalyst for the biodenitrogenation, biodesulfurization, and the bioupgrading of petroleum and its distillates, can be used as a biocides and water disinfection, can be used as a catalyst for the transesterification and production of another type of biofuel, which is biodiesel, can be used also for wastewater treatment as a photocatalytic treatment. Okay. As a conclusion, by applying the green synthesis and the nanobio-catalyst, excuse me, where is the slide? By applying the green synthesis or sustainable green synthesis of nanobio-catalyst and its application in different fields of biotechnology, you can have a great positive impact on national economy, environment, industry, and the energy sector, as you can produce alternative green fuels, reduce the dependency on foreign fuel, create green energy industry, produce green catalyst, corrosion inhibitors by site. You can issue less carbon dioxide and less emissions of the greenhouse gas emissions in general. And also decrease overcome the most of our waste management problems in a very economic and valuable way, preserving our food supply chain. They would significantly reduce unemployment, desertification, food problem, and open up huge markets for recycling and waste. Briefly, the valorization of waste, especially the organic waste, to useful, efficient biocatalyst and nanocatalyst and biocide agro-industrial waste can be valorized into inexpensive feedstock to biofuel. All of this will have positive impact on energy, environment, economic, waste management problems. Okay, so in conclusion, okay, upon recycling, the possibilities are endless. So there will be always a better tomorrow by thinking green and go towards sustainability and using of nanobio catalyst to have a green and sustainable environment, which will achieve the three pillars of sustainability, the economy, environment, and healthy. So, okay, and now thank you, and I'm ready for any question as long as you wish.
So I would like to ask the audience if they are having any query, please mention in the text box. We can surely ask ma'am to solve that issue. Lastly, I would like to thank Dr. Noor ma'am for providing us such an insightful lecture on such a significant area related to the field of green nanobiotechnology and environmental biotechnology. And I strongly believe that this insightful lecture will definitely help them to explore more sustainable solutions to several environmental issues, which ma'am has elaborated very well, and she has given a thorough knowledge about all the environmental issues and how to utilize the natural bioproducts for finding sustainable solutions to those issues. So I'm really thankful to ma'am, highly thankful to you ma'am, for providing such an insightful lecture on environmental issues, which we all are facing nowadays, ma'am. Thank you so much, ma'am.
And thank you for giving me this opportunity to join this wonderful conference. Thank you very much.
Now I would like to invite Dr. Fahad Khan, Assistant Professor in the Department of Biotechnology, to give a vote of thanks to all the eminent speakers who have graced their presence in this international conference. So I would like to now invite Dr. Fahad Khan to please provide the vote of thanks to all the esteemed speakers.
Thank you, Dr. Pratibha. I, Dr. Fahad Khan, on behalf of Department of Biotechnology, NIT Greater Noida, and on behalf of organizing team of this conference, extend a very hearty vote of thanks to the honorable delegates who blessed us with their presence and took out valuable time of your busy schedule. I would like to thank all the esteemed speakers, especially Dr. Pavandhar, Dr. Noor, and Dr. Zahra Iqbal for gracing your significant work and sharing with your findings and opinions which are in accordance with our theme of conference, that is advancement and challenges in science and technology. Lastly, I would like to thank Ms. Meenu Agarwal and Dr. Pratibha Pandey for hosting this technical session 3 of this conference and executing this session in a very smooth manner. Beside these, I thank Mr. Vikas and Mr. Amarpreet for smooth functioning of this technical session. I appreciate their work and thank you very much, all of you, for this session 3 of our conference. Thank you very much. We'll see you later in the next session. Thank you very much.